Hello, welcome to today's What's New in MG Research webinar. I'm Jen Umbalo, and I'm the National Director of Programs at the MGFA. Before we get started, I wanted to go over a couple of items. We want to give a special thank you to today's presenting sponsor, Arcellx. We're excited to have you here and for this information that we'll be receiving. Today's session will be recorded and available on the MGFA YouTube channel as well as our website following the presentation. If you would like to look at closed captions throughout the presentation, you can do so by clicking on the CC box located at the bottom of the Zoom icon and select English as your language. You can also submit your questions in the Zoom Q&A box located at the bottom of the Zoom menu bar, and we'll be able to answer those questions. With that, I'd like to hand it over to today's speaker, Dr. Chris Heery. Welcome.
Thank you. And thank you for having us here to talk through this topic of a new possible therapeutic that's being developed and evaluated in myasthenia gravis. So first, I'll give a quick introduction about me. Won't spend too much time on that other than to say that actually my background is as a medical oncologist, not an expert in myasthenia gravis. And so your obvious question might be, why are you talking to us about myasthenia gravis then? And it's because over the last 15 or so years, a technology has been developed that has had major impact in the field of oncology. And through understanding the physiology of the diseases that are within the same bucket of myasthenia gravis, autoimmune diseases, researchers have asked the question, is it possible that we could use these therapies to address some of the underlying causes of some of the autoimmune conditions that exist? And we'll talk about why they had that idea and why we think it makes sense as we go through the presentation. But my background and training as an oncologist led me to running clinical trials in patients with cancer. I did that at the National Cancer Institute for about 7 years. And for the last 9 years or so, I've been working for biotech companies. And my job in those roles is to help guide the clinical development of therapeutics to try to improve outcomes for patients with cancers and other diseases. And so in this case, we're applying some of what we've learned in oncology and some of the understanding of those therapeutics to this disease, myasthenia gravis. So just the agenda, just to give you an overview of where we're going, and you can probably track back to this if you are watching this on delay or watching it later if you want to skip ahead or back or forward, just to know where things lie. So we're going to start. I'll give you a quick overview of our company, why we are giving this presentation in the first place. Then I'm going to talk to you about this therapy called CAR T-cells. And to do that, we'll lay out what are T-cells in the first place, what is a CAR T-cell, how does it actually work, how do patients receive them, all of those things. We'll talk about our specific CAR T-cell, which we call anito-cel, and why we believe it is capable of doing what we're trying to do in this current clinical trial. And then we'll talk about the design of this clinical trial, and we'll answer any questions people have. So hopefully we'll leave about 15 to 20 minutes for questions. So Arcellx is a company that has a stated mission of advancing humanity by engineering cell therapies that are safer, more effective, and more broadly accessible. And those words may not mean much to you until we start talking about what cell therapies are and why there's a need to have safer ones and why they need to be more effective and what does even broadly accessible mean. But I'm going to give you just a foreshadowing of that. Cell therapies really are a broad collection of therapies where we are taking cells from a human being and modifying those cells or enriching those cells in a way to make them fit for a purpose to treat a particular disease. And then once we have them the way we want them, giving them to a patient. So really we started to see this field expand quickly in around 2009 and 2010. We were starting to see some early data from what we call CAR T cells, which we'll come back to in a minute. And those data were very impressive and even, I would say, surprising to the field, just how well they could eliminate cancer cells. But at the time, there were, significant challenges in terms of safety that were observed. And over the years, there have been efforts to understand why those safety events happen and to be able to prevent them or reduce the impact of those safety events, including modifying the design of these cell therapies so that you could mitigate the negative impact or safety risks while increasing the efficacy or effectiveness or ability to, in this case, kill cancer cells. And we've certainly seen that evolution over the last 15 years. The overall, just taken as a field, these types of therapies have now gotten to the place where they can be managed in academic medical centers quite well for the large majority of patients. And those patients, the large majority of those will also see some amount of measurable benefit in terms of reduction of their cancer. So, as we look at this field, it's not unusual in the field of cancer research or medical research in general to see a therapy start somewhere and then improve over time as we get to understand it better. But at Arcellx, one of the things we've done is tried to modify a certain component part of these T-cell CAR T cells, and I'll show you that later. And we think that particular component part offers some benefits compared to other CAR T cells. So we, as a company, are developing a range of therapeutics. And at the top here, I'll use my pointer so you can see we've been developing this same therapy, anito-cel, with our partners, Kite, which is a company within Gilead, in multiple myeloma. We've been doing that actually for a number of years and then partnered with Kite about 2.5 years ago for this program. That program is actually in the current stage, moving forward with a goal of getting this product approved for the treatment of multiple myeloma by the FDA. And our expectation is that this product will be approved and available as a product that can be given as a standard therapeutic sometime next year, sometime in 2026. But we're also developing therapeutics against other diseases. One is called acute myeloid leukemia. There are also tumors that we refer to as solid tumors like small cell lung cancer, hepatocellular carcinoma. And then here, in our non-oncology indications, our first disease that we decided to go after was generalized myasthenia gravis with this same product that we use in multiple myeloma that we call anito-cel. We'll come back to the why. Why would a therapy make sense for multiple myeloma and also generalized myasthenia gravis? But I think the reason I'm pointing this out is that we will walk through some of the data that we've seen in multiple myeloma so far. And those data are only really to illustrate that we have confidence that this therapeutic does what it's supposed to do in terms of eliminating certain types of cells. And why that's important, we'll highlight in some subsequent slides. So what is a CAR T-cell? First, we need to start with what is a T-cell, because for most people, you don't know what a T-cell is, and that's totally reasonable. Why would you? Within our immune system, there are a number of different kinds of immune cells that work together to help eliminate risks in our natural environment. For instance, if you have a bacterial infection, there's a type of cell called a neutrophil that will usually be one of the first to help eliminate that type of infection. But for infections that make it inside of our cells, we have to have a way to actually eliminate the diseased cell within our body. And that's what T cells are there for. So what is an infection within our cell? Well, the ones people would most know about are viruses. Viruses only work by infecting our cells and then making more of themselves within those cells. And then it actually kills the cell and releases more virus particles so they can go infect more cells. So there are actually multiple ways that our body helps to eliminate viruses, but one of them is to actually try to root out where that virus is by killing the cell that is infected with the virus. And so T cells really are sort of smartest immune cell, and that they only kill cells that are directly infected or have some sort of problem within them. And the way they do that is they recognize little bits of information, which are little pieces of proteins that are presented by the abnormal cell on the cell's surface. So this is part of our immune response. Our infected cells actually have a way to put things up on the surface and say, hey, this is inside of me. And then the T cell can say, that shouldn't be there. I'm going to get rid of this whole cell so that I can prevent this virus or pathogen, whatever it is, from infecting other cells around it. And so part of our immune system is actually eliminating infected cells. And that's what T cells do normally. But in cancer, it's not always true, for instance, that a T cell can recognize what's abnormal about that cell. It actually sometimes looks just like a normal cell. So over the years, scientists identified a way to modify the T cell so it would have these other ways of recognizing cancer cells. And so on cancer cells, there tend to be, on the surface of these cells, markers that indicate this is an abnormal cell. And in this case, actually, what we're doing is using a normal marker, a marker that's present on normal cells, but these are plasma cells. We're going to come back. This is another immune cell that is part of that disease that I mentioned, multiple myeloma. And on plasma cells, there's a protein called BCMA. And what has happened is we've taken that same T cell that's good at killing normal cells if it needs to, and then attached to it something we call a CAR. A CAR is a chimeric antigen receptor. That name may sound, confusing or overly technical to you, but really what you can think of it as is you added another attachment arm to this T cell that can recognize that target. And the reason we in multiple myeloma want to target BCMA is because plasma cells, which are the cells that express BCMA, are actually the cells that become the cancer cells in multiple myeloma. Now, plasma cells also have a very normal function within our bodies. And so BCMA is still expressed on the normal version of plasma cells. So if you made a CAR T-cell that recognizes this, this target antigen here, it would kill the cancer cells that express that antigen, but it would also kill the normal cells that express that antigen. And so that is part of the way CAR T-cells work. When we kill the multiple myeloma cancer cells, we also kill normal plasma cells in the course of treatment. Your next question might be, isn't that dangerous to kill plasma cells? And the answer is, it could be, but we actually have ways of managing that elimination of plasma cells in multiple myeloma. And because those plasma cells in multiple myeloma are abnormal, they actually don't really work as well as they should in the first place. So eliminating all of them just gives you a chance to reset the system. And that resetting of the system is going to be important. So remember that idea that eliminating all the plasma cells might allow you to reset things. That's part of the rationale that we're going to come back to for myasthenia gravis. All right, so a little bit of background about how we actually do this and how do CAR T cells actually, how are they made? How do they get to you? So what we do is we hook a patient up to a machine called an apheresis machine that actually allows the blood from the patient to flow out into this machine, and then the remainder will flow back into the patient. So you're not actually losing a lot of blood volume, but when it goes through the machine, it filters out these cells that we want, called the T cells. So apheresis is just a method by which we are going to hook you up to a filter and take out the T cells into a separate container. And then we take those T cells and we ship them to a laboratory where we insert a gene into that T cell. So what do I mean by a gene? So if you know what DNA is, this is the part of our cells that encodes all the proteins that make up what our cells do. And so we put a gene into your cells or the T cells, that gene causes expression of a protein. That protein is what I just referred to as the CAR, the chimeric antigen receptor. So that protein actually allows us to have this receptor on the surface of the cell that tells the T cell, this is what you need to go after, and this is what you need to kill. So we do that within the lab, and then we grow that up to hundreds of millions of these cells. And the reason we do that in the lab is we can do that under controlled conditions. We can make sure these cells are healthy. We can make sure that they're meet certain safety and quality attributes that we think are going to be important for the ability to do what they need to do and not cause safety risks. So that process of growing up the cells and making them actually only takes about a week on average. But then there's a bunch of things we need to do to make sure that those cells are safe to give to a person. And that process right now for this trial that we're going to talk about takes about 3 to 4 more weeks. Just for reference, in the therapies that are approved, and particularly this version of our therapy that will be approved in multiple myeloma, we are actually going to see those therapies from the time of the apheresis until the time it's delivered back, probably be closer to 2 to 3 weeks on average overall, where this is going to be more like 4 to 6 for this clinical trial. But that's more to do just with where it is in the stage of development and whether people need it as urgently. So in this case, there's not as much urgency to have these things turned around as quickly. So after those cells are ready and they're released to be sent back to the patient, they are shipped back to the site where the apheresis happened at your clinical trial site. And then in this study, we will give 3 days of low-dose chemotherapy. And the reason we do that is to actually knock down some of the other immune cells for a short period of time, about 2 to 3 weeks, so that the CAR T cells can do their job most effectively. In the future, we may or may not have the need for the chemotherapy in myasthenia gravis. That's still an open question. But for now, in this study, there will be this low-dose chemotherapy because we know from clinical trials in cancer patients that low-dose chemotherapy actually does improve the ability of the CAR T cells to work and work really well. So we've kept it in for now. Then after the chemotherapy, you have 2 days of rest, and then the CAR T cells are given after that. And then the CAR T cells go and do their job. There's nothing really else that we do to make them work. They go and find the cells that we want them to find. They actually can make more of themselves within the body, and their job is to eliminate the cells that they're targeting. So, that is not obvious maybe to a patient, but what is different about a CAR T cell is that you're not giving a medicine and then, taking a pill every day or coming back for an IV infusion every other week, which most medicines that's how it's done. In a CAR T cell, because of the way it works, it's an active cell therapy. We give it once and that's it. We don't give it again. And the reason is because it is so effective at killing the cells that it's supposed to, that there's really not a reason to give it again. In most cases, for patients with cancer, for instance, we only give these therapies once. And in myasthenia gravis, we believe that giving this once could result in long-term impact. And I'll come back to what we mean by that. These are personalized, so every product is made just for that specific patient. It's made with your own cells. It goes after, as I said, the specific root cause of the disease. And so in multiple myeloma, that's killing plasma cells because they are cancerous plasma cells. In patients with myasthenia gravis, killing plasma cells because plasma cells make the antibodies that cause myasthenia gravis. We'll come back to that in a bit. And so what we hope, based on what we've seen in the literature, is that these types of therapies may result in long-lasting benefit that allow patients to not need repeated doses of therapeutics. That is obviously still something we are evaluating, but there is reason to believe that that might be true, and I'll share with you in just a moment why we think that might be true. So what are the downsides of CAR T cells, at least that we've seen historically? In cancer patients, we've seen something called CRS. This refers to cytokine release syndrome. So cytokine release syndrome is a descriptive term. When these cells are killing the cells they're targeting, they will release these things called cytokines. Cytokines are really just signals between cells. So in this case, the T cell, as it starts to kill, is trying to tell other immune cells that they need to come in and help do their job. And those cytokines, when there are a lot of them, can actually make you feel sick. So actually, a lot of times when we have the flu or if we have an infection of some kind, what we feel in terms of fevers and chills and all these things are not actually the infection. It's actually our immune response to that infection that we feel. And that's mediated by the cytokines that our cells are releasing to get the rest of our immune system activated to come and eliminate the infection. And that happens with CAR T cells. In myasthenia gravis, we actually don't expect cytokine release syndrome to be quite as severe as it can be in cancer patients, and I'll talk about why that is. Now, we do not know that for sure. I want to be clear about that, but we believe that these may be relatively mild symptoms for some very specific reasons that I'll go through in just a second. Similarly, in patients with cancer, we have occasionally or we do sometimes see an entity called ICANS, which is immune effector cell associated neurotoxicity syndrome. So what does that actually mean? It means that some of the patients can have confusion, delirium. Usually these are temporary, usually lasting a few days. And these are almost always when we see it in cancer patients, they're almost always an effect that is preceded by cytokine release syndrome. Not every single time, but it's usually associated with it. And you can think of ICANS or this neurotoxicity delirium as sort of on the same general spectrum as cytokine release syndrome. And so it's sort of part of the whole continuum of events. We also do see that patients after the lymphodepleting chemotherapy can have low blood cell counts, meaning some of the normal immune cells, and then therefore can have risk of infection. So let me come back to some of those things in more detail in a minute. But I do want to explain why we believe that these adverse events may not be as prominent in autoimmune diseases as they are in cancer. So in cancer, what we're showing here is the purple cells are the plasma cells, the cells that we're trying to eliminate. And because it's cancer, we actually have way more of them than we should have in our body, right? So in the case of multiple myeloma, you're going to have billions and billions of extra plasma cells because that's the disease. They made more of themselves and they're causing problems. So when the CAR T cells come in, there have to be more CAR T cells because they've expanded to eliminate these cells. And so they make a lot of cytokine, which is the little dots you see in the back. Now, in autoimmune disease, we think the relative quantity of plasma cells should be significantly lower. This should be the normal amount of plasma cells in a patient's body. Now, they're making abnormal things. They're making antibodies that cause myasthenia gravis, but there are less of them. So you don't need as many CAR T cells in the expansion phase, and therefore you don't have as much cytokine floating around. So it stands to reason, based on this hypothesis, that higher disease burden patients will have more cytokine release syndrome, and lower disease burden patients have lower cytokine release syndrome. And you'll be happy to hear that that actually is true in patients with cancer. So patients with cancer have higher disease burden, they have more cytokine release syndrome or are more likely to have severe cytokine release syndrome. And patients with low cancer burden actually have way less. So this logic is backed up based on research and observations in cancer patients. But I should clarify, we still don't know this for certain about myasthenia gravis. It's just our interpretation of the data that we've had so far. To give you a little bit more reference for this, we can look across therapeutics that have been used both in autoimmune indications and oncology. So for instance, within these programs, you can see that higher grade CRS, and that means more severe, did not happen in the autoimmune program from this company, BMS, but it did happen 3% of the time in patients in their oncology setting. And then similarly, in this company, Novartis, 0% of the time in patients with lupus, another autoimmune disease, versus 6% of the time in the oncology program. Similarly, for that, confusion ICANS that we were just talking about, the rate of those events is only 13% in autoimmune disease for this program versus 31% in the cancer program, and 0% versus 25%. So you see there's a difference between the two. And that's very much backed up by the observations in the cancer studies related to the total burden of cells that we are trying to target. All right, so now the question for some of you might be, then why are you trying to eliminate these cells at all in myasthenia gravis? So as some of you probably know, if you've been learning about your disease or your friend, loved one's disease, what myasthenia gravis is the production of abnormal antibodies. This is another part of the immune system. And what antibodies do is sort of latch on to things that they want to neutralize or need to eliminate. And in this case, the body is making antibodies against something normal in our body. So that's what we mean when we say autoimmune, where our immune system is attacking our normal part of our body. These antibodies are binding in the junction between our neurons and where they synapse with the muscle. There's a couple of different kinds. We're not going to go too deep into that today, but the point is that these antibodies are made predominantly by a cell called a plasma cell. Plasma cells are sort of the end version of another type of immune cell called B cells. We're not going to spend a ton of time on that either today, but just know that plasma cells actually make the large majority of antibodies in any person. So it stands to reason then that if you could eliminate with a CAR T-cell, if you can eliminate that cell, the plasma cell, then you would also eliminate the production of those antibodies and therefore could prevent the damage that they cause in the synapse between the muscle and the neuron. So I'm going to pause there for everyone to just try to catch up with that thought, because it's a key thought. We, in this case, are trying to attack the cell that makes the antibody that causes the problem. And by doing that, our hope is that if we eliminate these cells, enough of those cells, that they will actually reset. And when your body then makes new plasma cells, they won't be making these antibodies anymore. Now, I want to be clear that that is a hypothesis at this point. We do not know for certain that that will be true, but there are reasons to believe it could be true, which I'm going to give you a couple of examples that we've seen in the literature so far. So these are two patients who were treated on a clinical trial in China, both with myasthenia gravis. The first of these patients, they're outlined here, and I'm not going to go through every part of this because there's too much information for this discussion. But the first patient had one of these antibodies and had generalized myasthenia gravis with weakness in their limbs and in their breathing. The second patient had a different antibody, but the same impact in terms of causing myasthenia gravis, and was having crises 1 to 2 times per year, had difficulty chewing, swallowing, and walking. Both of these patients received a different BCMA CAR-T therapy. And then after that, they had significant improvement relatively rapidly. So their scores by MG-ADL, they were down to 0, meaning they didn't have any symptoms by 3 months after treatment. And so it's really important to think about, like, what does that translate into? It meant those patients were not receiving any other therapy and were able to return to their normal function. And that's really why we're running this clinical trial. We want to see if we can do that and potentially do it even better than some of the other CAR-T products out there. I'm going to come back to why we think our product might be better at doing this than others. But it's important to also mention that in terms of safety, even though I mentioned the CRS, et cetera, the first patient had that CRS syndrome, which is really just a fever that lasted just 1 day and then resolved. And the other patient, was sorry, neither of the patients had the delirium, confusion, ICANS event that I mentioned to you. Now, one of the patients did have an infection 10 weeks later, but what we hope is that after these therapies, the immune system will actually recover better than it was on the immune suppressive therapy that is required for myasthenia gravis, and that months out, the risk of infection will actually decrease over time. Again, that's a question. It's not something we know for sure, but that's one of the questions we hope to answer in this trial. So what does our study look like in myasthenia gravis? It's a phase 1 trial. And you might say, well, aren't phase 1s to find out if something is safe? And the answer is yes. And does that make sense in a therapy that we've already put through multiple stages of development, phase 1, phase 2, and actually this is in phase 3 now in multiple myeloma? And the answer of why we call this a phase 1 is because it's in a totally new indication, and we need to find out what the safety profile looks like in myasthenia gravis. We actually expect it to be different than it was in multiple myeloma for the reasons I've already talked about. So it's called a phase 1, but you should rest assured that this is a therapy that has actually a pretty long track record now of being in people. We already know a lot about the therapy, and people know what to expect, know how to manage certain things. So I mentioned already that you start out by screening. That's to find out if you're eligible for the study. You go through that leukapheresis or apheresis that I mentioned earlier to have your T cells taken out. They go to the lab to be made. This is that manufacturing. Patients are allowed to continue on therapy to control the symptoms of their myasthenia gravis while waiting to get their treatment. And then, like I said, about 4 to 6 weeks later, those cells will be back. You'll stop your therapy for controlling the disease. You'll start the low dose chemotherapy, and then 2 days after that ends, get the infusion of the CAR T cells. After that, it's really just following up. And we ask for patients to be in the hospital for this because we're just learning about this disease at this point with this therapy. So we want to be extra cautious. So patients will be in the hospital to be observed, to be monitored. If there's anything that needs to be intervened upon, we have already written out protocols for how to manage those situations. And we are in communication constantly with the doctors who would be taking care of the patients on this study about what we've learned in the past and how we can help manage things best. And then we follow the patient for a long time, up to 2 years. And then beyond that, even we would follow up with less formal interactions, but we want to know how patients are doing. But what we hope is that this will result in long-term disease control without the need for other therapies. So what we're hoping to see is patients can go a year or 2 years without needing other therapies. And that's why we want to follow patients for a long time. We have explanations of some of the things we need to do to follow. So things like adverse events, that's what AE stands for. DLT stands for dose-limiting toxicities. And then measuring things like, did your myasthenia gravis scoring scale improve with therapy? So did that improve after treatment compared to what it was before? So there are a number of them when you participate in a clinical trial. There are a number of things that you're going to be asked to do. And some of them will feel like, gosh, that's a bit of a pain. It's a bit of a hassle. But we're doing it because we need to learn these things and we need to collect that information prospectively so that we can apply it to what needs to happen for other patients. So participating in a clinical trial can be very rewarding for patients, but it can also be a hassle. And we understand that. We try to minimize the hassle as much as possible. But anyone who chooses to participate in a clinical trial should be informed of those things and should make that decision knowing what they're getting into. So if you're not sure what you're getting into, make sure you ask if you're considering this or any clinical trial. But anyone who does choose to participate in this study or any other study, I personally thank you. We wouldn't see the improvements that we see in medical care over the years without patients being willing to participate in these studies. So thank you to anyone who's considering it or actually does participate in this or any trial. I already mentioned what happens in terms of cell manufacturing and things. And this is just an overview of what to expect. I'm going to leave this here for a second again so that you can pause it and read through this if you'd like, but I'll just remind you the apheresis followed by making the cells, followed by infusion of the cells, followed by the pre-treatment low-dose chemotherapy, followed by the infusion of the cells, and then following up. And we start out with more visits, more frequent visits at the beginning, and then they spread out over time. And that would all be, if you're interested, in the informed consent document that you can read about the study as well. Right now, we have this study open at a variety of centers around the United States. We're not in any other countries with this therapy right now or this clinical trial right now. So we range all the way from the Pacific Northwest down to Texas and all the way back up to the Northeast. We are working on adding some additional sites. We know that for patients, it can be really difficult to travel far from home to participate in trials like this. As you can imagine, it's somewhat helpful to have these sites open where there are lots of people. And so these tend to be where larger cities are, as you can imagine. So for anybody who's interested, if you live further away and would like to participate, but you're worried about cost, we do offer financial support and or reimbursement for costs for meals, travel, overnight stays to participate in this trial. We don't intend for that to be an incentive to get you to go on the study. We just want to reduce the impact on your life if you want to participate, but you otherwise can't afford to make the trip. So please consider that. If you need more information, or if you're watching this weeks from when I'm presenting this, and you want to know what other clinical trial sites are open, you can click on this clinicaltrials.gov link, and it should take you to a website that at the bottom will tell you what clinical trial sites are open. So I'm going to just quickly summarize, and then we'll go through questions. What I've told you today is that CAR T-cell therapies are an engineered version of your own T-cells. T-cells are the type of cells that eliminate infections that are within our normal cells. CAR T-cells are modified so that they can recognize a specific cell type and eliminate those very efficiently. One of those CAR T-cell targets is called BCMA. BCMA is present on cells called plasma cells, and plasma cells are cells that produce antibodies, and antibodies are what drive the disease of myasthenia gravis. One BCMA CAR-T, which is the one we develop at Arcellx, is called anito-cel. Anito-cel actually has a very strong track record of being efficacious and relatively safe in a cancer indication called multiple myeloma. And we are now evaluating anito-cel to see if it can do what we think it can do in myasthenia gravis, which is eliminate plasma cells and do it relatively safely, and then hopefully have long-term disease remission in patients. And that's the big questions we're asking in this clinical trial. So if you are interested in participating in this clinical trial, please reach out to one of the clinical trial sites that we just had on the screen, and you can find their contact information by going to this website. If you have any other questions, you can also reach out to the MGFA, and they can help get you connected to the right sites as well. So I'm going to pause there and look through the Q&A and see whether we can answer some questions. First question, which is a really specific one and a good one. Will this work if you have had a thymectomy? So as some of you may be aware, a thymectomy is one of the potential interventions that is used for myasthenia gravis. And so whether or not a patient has had a thymectomy really doesn't have any impact on your eligibility for this trial or the likelihood that this mechanism could work that we know of. So thymectomy is not an exclusion to participate in this trial, and we don't believe that it should have any impact one way or the other. Now, I should add one caveat, which is if a patient had a thymic carcinoma or a thymoma, that is something that we do need to look into when we're considering eligibility for the trial. So I just want to make sure if the thymectomy was for a malignancy, that might be different. If the thymectomy was primarily for treatment of myasthenia gravis, no impact on eligibility or what we believe the mechanism of this therapy is. Next question was, why does the therapy target plasma cells rather than cells further upstream like autoreactive T cells and activated B cells? It's a really great question. This is a little deeper in the weeds for some of the audience, I'm sure, but actually the cascade of events that leads to immunity at all, and probably most autoimmune diseases, is that initially there has to be recognition of an antigen by some cell part of the immune system. So typically that's going to be an antigen presenting cell is going to teach a T cell, it's going to show something to a T cell, and the T cell is going to recognize that and then go after that target. Why that happens is complicated background. I don't think we have time to go into that, but the question is really getting at why don't you go all the way back to that root? It's a really interesting question. And so the challenge is that you, in order to do that, would probably either need to eliminate all T cells, or you would have to know which specific T cell is driving that autoimmune reaction. And in myasthenia gravis, I don't know that we know that. But what we do know is that those T cells eventually lead to activated B cells, as the question asks. And then eventually those activated B cells lead to terminally differentiated plasma cells that make the highest quantity of antibodies. And so it is possible that, and it is likely even, that BCMA is partially expressed at some level on some of the activated B cells, and may help to eliminate that. It's also possible that whatever the trigger antigen was is actually no longer present or no longer being presented at the time a patient has symptomatic myasthenia gravis. And so eliminating plasma cells may eliminate the last remnant of that recognition. It is a good question, and it is part of the reason we have to run this clinical trial to know the answer to that question. So mechanistically, if we could, would we love to get all the way back to that root cause of the recognition of the antigen? Yes, that would be the ideal. But that's a technically very challenging solution, where this solution may be much more practical and still offer the same clinical benefit. The next question is, are long-term side effects known? And is there any data from this and from the cancer patients' experience? Yeah, there are some. In cancer patients, occasionally we will see patients who have done really well with the therapy develop other cancers. And one of the things we have as an open question in this field is that related to the CAR T-cell, or is it related to all the therapies they received for their cancer before they got the CAR T-cell? And the jury is still out on that. If you look at this from a pure data standpoint, you would say CAR T cells are associated, relatively rarely, but are associated with the risk of a secondary cancer. But if you look at the same group of patients with cancer who receive CAR T cells, many of those patients would have already died if not for the CAR T cell. So it's really hard to answer the question definitively whether the CAR T cells caused the cancer or whether they allowed the patient to live long enough to have another cancer. We don't know whether that risk is real or the same in other indications like myasthenia gravis, but it is something you should be aware of as a theoretical risk, and something that will be present on the informed consent form if you're considering a trial like this. The next question is, if CAR T targets plasma cells, do these people need to be revaccinated with all the vaccines they received in childhood? Great question. We would recommend that. Yeah. So once the plasma cells, normal plasma cells, start to repopulate, which we have seen in other BCMA CAR-T trials, tends to be around, somewhere between month 6 and month 12 after therapy. You definitely should be revaccinated during that window. Most people actually get revaccinated earlier than that, but certainly would recommend a revaccination strategy, even if the literature suggests it's not necessary, which some of the literature suggests you may not need to be revaccinated. But if it were me or a family member, I would absolutely go through a revaccination strategy for really common diseases, just to be safe on those things. There's no reason not to. Next question was, in the 2 patients in China who went into remission, how many others were in this experiment? That is a good question. I don't know the answer to that question. This was in a published paper, and those are the types of things I would like to know as well. So great question. But it is exactly why we are running this clinical trial, because we would like that level of understanding and transparency. We want to know, if we treat 10 patients in a row, do all 10 of them do well? That to me is a much more reliable way to report information. And unfortunately, I just don't have that answer for you about that study. Next question is, are there different plasma cells for each type of antibody? That's a really good question. Yes, the most likely answer to that question is that for each specific antibody, there are probably clonal plasma cells that make that antibody, and that they react to the presence of an antigen when it comes back into your body. So, for example, if I've been exposed to influenza And my body has learned how to make antibodies to help neutralize influenza virus. And my T cells have learned how to kill cells that are infected with influenza virus. The next time my body sees influenza, the plasma cells that make influenza antibodies are going to become reactivated more rapidly and make more of that antibody quickly. And then when the flu goes away, then the plasma cells calm down again, right? So that's why, for instance, when you get a vaccine, the day after, if it's a vaccine you've had before, you tend to feel kind of sick because you're getting essentially, remember how I described cytokine release syndrome? You're getting sort of this activation of the immune system. It's not that you're actually sick with an infection, it's that your body is responding to the signal that it just got. So yeah, there are probably different plasma cells for each type of antibody. And the idea here is that if we can eliminate a lot of the plasma cells, we may eliminate the one that is specific to the myasthenia gravis antibody. That's a really great question. Next question, is there a way to ever turn off the CAR T cells in your body? If they never turn off, then theoretically we could potentially always be in remission. That's a really great question. I have to admit, I am very impressed by the level of questions you all are asking. So CAR T cells, there is a debate, and I'll just acknowledge this debate, that some people believe that CAR T cells stay in our body for the rest of our lives and help to eliminate the target cells forever. And then there are other people who believe that those CAR T cells go in and they eliminate the target cells, but after a while, they get worn out and they just can't keep going forever. I will reserve my opinion on that, but I will say that there are data that indicate that even after a patient has received a CAR T cell, they recover the cells that the CAR T target, right? So what does that suggest? It suggests that after some period of time, the CAR T cells cannot continue eliminating the target cell, and that those target cells can actually return to normal. We see that in multiple myeloma, and evidence of that we see antibodies recover in patients with multiple myeloma after a period of time, after therapy. And so we have to wait and see what happens in patients with myasthenia gravis, but our expectation is that patients will recover normal plasma cells and that those normal plasma cells will make normal antibodies some period of time after treatment. We don't know exactly when that will be, but if we go by what we've learned from multiple myeloma, we would think it'd be somewhere between 6 and 12 months after treatment. So while it is a nice way to think about it, hey, could we be in remission forever because of that? I'm not sure whether that will be the mechanism. It may be because we eliminated the plasma cell that makes the bad antibody and that it never comes back. But I don't, maybe I'm giving away my opinion, but I don't think it'll be because the CAR T cells are there forever, eliminating plasma cells. All right, next question is, how many days does the chemotherapy phase last? So it's 3 days. It's from the 5 days before you would get your cell to the third day before. So it's day -5, -4, -3 before the cells are infused. And it's the same dose every one of those days. Next question is, if you're doing rituximab infusions for 1.5 years every 6 months, would seeking to do T-cell therapy be better? Can you participate if seronegative? The patient was diagnosed 2 years ago, has remained with ocular symptoms only. Yeah, so these questions are very specific about eligibility, and I would encourage, if you have specific eligibility questions, please reach out to one of the sites, and then we can have the site go through the eligibility review with you one-on-one or reviewing the records. That would be a better way to do this. Unfortunately, I'm not really able to comment specifically about individual subjects while we do this. I think, I don't want to cause confusion in doing that. So next question, how fast are the plasma cells replenished after being depleted by BCMA CAR-T? That I think I've already answered that, hopefully, and with the best guess that we have, somewhere between 6 and 12 months. If you eliminate plasma cells, are you able to fight infections? So yes, you can still fight off other types of infections, but you will not be able to produce normal amounts of antibody for some window of time. So you'll be able to fight, like, for instance, the cells that fight off bacteria, neutrophils, they should recover, on average, they recover less than a month later from treatment. on average, your T cells will recover less than a month later from treatment. And those are the cells that fight off intracellular infections. But what you won't have is the ability to make as many high quality or high concentration of antibodies in response to a new, say, viral infection that helps neutralize that virus when it comes into the body for the first period of time, maybe 6 to 12 months, something like that. Again, we're running the trial to find out the answer to that question, but that is one of the risks that you are more prone to these opportunistic type of infections during that window of time. Next question is, can I use this therapy if I am in another clinical trial for MG at the moment? No, we don't allow patients to be on this clinical trial while you're on a clinical trial of another therapeutic at the same time. That's just more of a challenge of controlling the information and what the impact of each therapy is. So we try to separate those out. Next question is, are there any specific requirements of immunizations? Again, you could probably best answer that question if you're interested by reaching out to one of the sites. But in general, there aren't immunization requirements necessarily, but we do want people to be prepped well to do well with this type of therapy. So we don't want, to have someone who will add to their risk by not having the right immunization background. So just keep that in mind. It's generally not advised to not have built up your immunity against known pathogens if you're also dealing with an autoimmune disease in general, or if you're getting a therapy that could suppress the immune response. Next question, what happens to the immune system after all the plasma cells are killed? I think I've covered that. Those plasma cells we do expect will repopulate over time, but there will be a window of time where you have less function or no function of plasma cells for a period. How is anito-cel dosed? That's a great question. So anito-cel is given as an IV infusion. It takes usually under half an hour to infuse it, more like 15 minutes, I'd say, for most patients. It's a bag. It's a bag of what looks like fluid, but it has cells in it. And we dose the number of cells based on where we are in the dose escalation strategy. So there are various doses, but the dose that we've used in multiple myeloma is 100 million cells, which sounds like a lot, but you can't see them in the bag. And the question, one of the questions in this trial is, do we need a higher or lower dose in myasthenia gravis? So, but the starting dose right now is 100 million cells. It may go up or down depending on some things we see in the study. Next question is, what about seronegative? I should have answered that part before. For seronegative right now, we are not enrolling patients that are seronegative, mainly because we are still testing the mechanism of action and want to be able to demonstrate whether anito-cel does what it's supposed to do. And the best way to do that is to measure an antibody, to have an antibody to measure. In the future, we hope to be able to go into patients that are seronegative, but only after we've learned the things we need to learn about anito-cel. So that may come in the future, but right now we're not aiming at patients that are seronegative, just for clarity. Will this be targeted toward other autoimmune diseases? Great question. We may in the future. So particularly autoimmune diseases that are caused by antibodies that are produced by plasma cells. So that is something we are always thinking about and looking at as a possibility, but I don't have anything specific to say about that at the moment. Next question, if you have low T cell counts because of taking immune suppressors, do you have to stop taking them before CAR T? That's a really good question. Yeah. So it really depends on what the medication is, and we do have very specific answers to that within our clinical protocol that the doctor that you reach out to could help answer. I can't answer those specifically on this call, but there are certain drugs that we do want to avoid for a period of time before doing the apheresis to make sure that the T cells are healthy. So it's a really good question. Next question, is there a potential that the activated B cells will produce plasma cells in the future? Yeah, I think that is a theoretical potential. So, coming back to this question of why not go all the way back to the beginning, that is a potential that a different target, CD19, may address, where we are addressing the cells that make most of the plasma cells. I think there may be a path for both, but that is a theoretical possibility. So that's one of the things that we hope to answer in this clinical trial. Are the patients in crisis or can they be in remission? What is the length of hospitalization? Yeah, these are some really good practical questions. So we really can't have patients like actively in crisis going into the treatment here. We do need patients who are under relative control, although they can be breaking through their current treatment. But we can't, unfortunately, have patients who are, not functionally capable of doing their activities of daily living and things like that. So there's a balance, and the inclusion exclusion can be found on that website that I shared on the slides. You can find that there, or by reaching out to the clinical trial sites to get that level of detail. In terms of hospitalization, when we dose the patients, we do ask those patients to remain in the hospital for 2 weeks if they're the first patient on a given dose level. In the future, that requirement may become less, but that's really primarily for safety. We just want to be able to monitor patients closely and make sure that they're being closely watched. Next question is, how does your therapy's mechanism of action differ from other complement existing treatments? Yeah, it's a really good question. I probably don't have time to go through that in detail, but a lot of the other therapies either block what the antibody does, meaning they actually create an interaction with the antibody to prevent it from binding, or they help eliminate that antibody. Some of the therapies also helps just suppress the cells that make some of the activating signals like T cells. So there are a variety of different therapeutics in myasthenia gravis that work different ways. And unfortunately, probably don't have enough time to answer all of those. I see we're running out of time. I will answer an important question. Someone said, are you actually recruiting? The answer is yes. We are actively looking for patients on this clinical trial. The best way is to reach out to those sites, and they can tell you when slots are available. But we are actively looking for patients who would be eligible for this trial right now. So if you're interested, please reach out to one of the sites, the closest one to you. If you need help with travel arrangements or whatever, please let them know that, and they can try to facilitate that. So I'm going to stop there and thank MGFA for having us today. Thank you all for being an amazing audience and asking really, really insightful questions. Like I said, super impressed with the level of questions. And hopefully we can do this again sometime. If there are follow-up questions, I'd be happy to do a follow-up. So thanks again.
Yeah, thank you very much. This was a really informative session, and thanks for taking the time to answer so many questions for us. We'll be posting this on the website shortly and sending out an email as well. Thanks, everybody, and have a great rest of your day.